All right, now for the last section of this chapter. Hopefully, you're feeling a little bit more confident with your chapter six, part two by now. Um, I'm going to go into basically some miscellaneous stuff with cellular respiration, and that's what the rest of this chapter is about. So they cut this section out of the newest book, and then they added that thing about, they put in the stuff about the different types of fat, the difference between white fat and brown fat. But I like this part, so I kept it in my PowerPoint. And this is how some poisons work. And the reason why I like it, not that I'm fond of poisons, but it actually kind of further shows how oxidative phosphorylation works and what happens when it's not working. So, for instance, this rotenone. So, rotenone is a type of poison, and what it does is it blocks the passageway of the electrons into the ETC. Therefore, we cannot make enough ATP. I mean, we do at least get through this first pump, but again, not enough to make that good proton concentration gradient to make enough ATP to keep the cell from basically self-destructing, right? It cannot maintain homeostasis without energy. It cannot oppose entropy. Cyanide and carbon monoxide. Now, you guys have heard of those chemicals, and you can see here that it completely blocks the handoff of that last electron to oxygen. So, if I cannot hand that electron off, we end up with a traffic jam and we cannot hand off the electrons. And so it just stops, it all backs up. And so, once it's all backed up, we cannot do our proton pumps. And again, no ATP is made. Well, DNP makes the membrane leaky. This is a very special membrane. I mean, think about how small a proton is and it cannot get through. And if it does get through, then we have this leaky membrane. We cannot establish our proton concentration gradient. Therefore, we do not get chemiosmosis and no ATP. And then the last one they talk about here is this oligomyosin, and it blocks the ATP synthase. So again, it doesn't matter if we have this proton concentration gradient, it can't get through. And if it can't get through, nope, no ATP again. So these are all ways that this can go wrong. And so hopefully if you understand the way it's supposed to work, if when I talk about what are some possible ways that is not working, you understand this process, which is what I'm going for here. So what happens if there's no oxygen? Okay, I already said, well, if there's no oxygen, we can't hand off to oxygen, everything backs up. But did we need oxygen in glycolysis? I mean, we made a couple ATPs, but did we need it? So let's think about that. So here is basically our glycolysis, right? We took glucose and we made pyruvate. When we went in this forward direction, we made two ATPs, right? And we also reduced our two NAD pluses to give us NADH. Well, if everything is backed up because you don't have oxygen, we do have a backup plan. All right. And this backup plan is temporary, it is a short lived thing. You cannot do that much with this plan B. So what happens is NADH says, okay, I'm going to just drop you back off. So it puts it back on pyruvate. It says, here, take your electrons back. And when it does that, see, I even put it on do not disturb, and we still got disturbed. When it does that, all right, so notice here, right here, I have two hydrogens. I don't have hydrogens here, right? Because I had to put them back on. And when I do that, I form lactate. You guys have heard of lactate, it is lactic acid. And so this is what we use in that fight or flight situation where, have you ever been winded? That's called oxygen death. Okay. If you've ever been huffing and puffing, which, you go up a couple flights of stairs and woo, and you're huffing, that means you have gone into oxygen deprivation, which means you were not able to sufficiently give enough oxygen to your cells for the needs of the ATP. All right. So your activity exceeded the rate at which you could supply oxygen to the mitochondria to keep up with its ATP needs. And so, what we're going to do here is we're going to try to squeeze out a little more ATP by forming lactic acid. And this is why maybe the next day after you worked out, that's that soreness that you feel. And that soreness, because it is an acid, tends to irritate the muscle fibers. And so you need to work that lactic acid out of your system, which is why coaches and trainers will tell you after a very hard workout, make sure you do something tomorrow. Because usually if you don't, day 2 is worse. And what happens is that lactate, we don't waste, we never waste. So that lactate needs to get from the muscle back into the bloodstream, and it'll do that by muscle contraction. So the more you do something on the next day, it helps to get that lactic acid out of the muscle fiber into the bloodstream and back to the liver, and the liver will then convert it back to pyruvate, and then it will go on its way through intermediate step and Krebs cycle. Right. So this is our backup plan if we are low on oxygen. But like I said, we cannot use that forever. And you see when athletes start cramping up or hitting the wall, that means they can't, they're done. We don't have enough ATP, even with fermentation here, lactic acid fermentation. You cannot keep this up. Now, some cells undergo what's called alcoholic fermentation. Now, it's the same concept, Glucose, pyruvate, right? We took off because glycolysis, all your single cell organisms, they do glycolysis also. This is a metabolism that's been around, that was around first in our prokaryotes. And so we make our 2 little ATPs, and think about it, bacteria don't need much energy. So this works just fine for them. And we're going to put them back on. So notice here, they're not here. We're going to say, take it back. And when it does, notice we have one here and here, but it's going to do this a little differently. We're going to lose this carbon dioxide up here. So we're going to lose carbon dioxide, and then we're going to make ethanol. That's ethyl alcohol. That's your alcohol product. So this is why it's called alcoholic fermentation. If you've ever made beer or wine, you know that basically your glucose is your sugar, like in your grape juice, it's high in sugar. You put it in an environment where it's in a sealed container. The yeast will use the oxygen up through aerobic respiration. Aerobic means with oxygen. Once the oxygen's all been used up, we now have an anaerobic means without oxygen environment, and then it will start to kick over into alcoholic fermentation. And then my dad used to make wine in the basement, and so he'd stick a balloon over the jug, and then the balloon would start to expand as it filled up with carbon dioxide. And that's when he could kind of tell when it was done. Yes, it was probably the most disgusting homemade wine ever. And now we are much more sophisticated in the kits that you buy today than they were back in the 70s. So here, our byproduct is ethanol. Now, ethanol is not dangerous for single cell organisms. And I'm going to talk a little bit about that. So without oxygen, we can make a little bit. So fermentation, the job of fermentation is to get a little bit more energy. Hopefully enough energy to complete our task. Maybe we're trying to catch dinner back in the day, chasing after that deer or a rabbit, or avoid being dinner or being chased by a bear. Okay, so trying to, like I said, our fight or flight response, trying to finish some kind of task to help keep us alive. We really don't have that today. Now it's like, can you make it to that finish line in that track event? Or can you get that last shot on a basketball court or run in for that final touchdown? I mean, sports are probably the closest thing that we have where we are going into that oxygen deprivation and trying to get a little bit more out of it. Now, my question for you guys is, why do animals use lactic acid fermentation instead of alcoholic fermentation? Now, if you think about it, and I don't know if I didn't think so. If you think about it, what does alcohol do I'm trying to get, jeez. Here we go. There we go. All right, I'm just going to forget that because I'm getting frustrated here. I'm just going to touch it once. Okay. If you think about it, single cell organisms do not have nerve cells. Okay. Without nerve cells, we know what alcohol does to nerves. If you don't, you will learn in anatomy and physiology. It impacts the way nerve function works. Okay. Most of you guys are over 21. You know what it's like when you drink. So, would it be a good idea if when you are chasing that bear and you go into lactic acid fermentation, instead of that, you go into alcoholic fermentation, which means your cells would start producing alcohol, which would then impair your nerve function, which then you might think, oh, that is a very cute bear. I want to give it a hug, and you die. All right. It might make some sports, those of us that don't like to work out, maybe it would make running much more enjoyable. But again, it would not be successful for us to produce alcohol when we went through oxygen deprivation, not to mention it would just totally kill our liver. So that process does not work in animals with nerve systems. So obviously that would have been a big evolutionary failure if that process was tried, and it might have been tried, but then it wasn't successful. And so individuals that were able to instead make lactic acid were successful, and that is the type of anaerobic respiration we see now in the kingdom Animalia, which has nerves. All right, alternative uses of cellular respiration. Now, we can obtain energy from other molecules. So we do get energy. Matter of fact, many of us are on these low carb diets. And what we're eating is foods that are high in fat or proteins. So I want you to pay attention to the next couple of slides. They're going to be anabolic and catabolic reactions. So the first one that we went through, we have our food, and this is what we did. We went from our carbs to our glycolysis to pyruvate to citric acid to oxidative phosphorylation, and we made our ATP. Now, we can also do this with fats. Remember, fats are triglycerides. And so they have that 3-carbon sugar backbone. And so that 3-carbon sugar backbone, that glycerol, right? Glycerol is right there. That glycerol can then go into where we have that 3-carbon, right? Where we have that 3-carbon molecule in glycolysis, that G3P. I didn't really give it a name, but it's there. It's G3P. And then it can proceed through intermediate step and all that good stuff. Now, the fatty acids, what we tend to do is break them into 2-carbon. Because remember, fatty acid tails lots and lots of carbons and hydrogens, and we can harvest those hydrogens. So we're going to break that molecule into 2 by 2 by 2, and then we can attach it to coenzyme A, and it can be escorted into the citric acid cycle, and where we can harvest those electrons from the hydrogen. Fats are full of hydrogens, which is why they have so much energy in them. Now, proteins, remember, we have 20 different amino acids. So depending on the side groups, the first thing we do need to do is a process called deamination, where we're going to pull the amino group off. And then depending on what's left, it could go in any one of these points to make ATP. So this is all breaking down. We're breaking down these molecules. So that would be catabolic. Breaking it down. I was thinking of catastrophes, break things down. All right, we're going to couple, because this is an exergonic reaction. We're going to couple that into this series of steps to make ATP. There's that energy coupling we learned in a previous chapter. Now, we could do the opposite. So if we need proteins or we need fats or we need carbs, we can pull these intermediate molecules out and synthesize or make them, which is anabolic. That means to build. So that would be an anabolic process. And so the last thing here when we talk about metabolic pathways, but often because we have all these intermediate substances here that we call them intermediates, they can behave for feedback inhibition. So if I have too much G3P, we'll stop there. If I have too much pyruvate, we'll stop there. If I have too much citric acid or succinate, or so remember, they all have enzymes and they can inhibit the next series of reactions. So there are ways for your cells to regulate the speed at which these chemical reactions or metabolism is happening. Okay, so that is the end of chapter 6. I know it was difficult. Again, hopefully this last section was a little bit easier to understand. But as always, if you have questions, please feel free to reach out to me and I will do my best to answer them.